Today I'm going to give you a quick introduction into genetic mutations. But first, let's go over the central dogma of molecular biology, which is just the idea that genetic information in a cell is stored in the form of DNA, and this DNA is used to generate complementary RNA through a process called transcription. That RNA is then used to synthesize a corresponding protein through the process of translation. So looking at a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with U, or T, and C pairs with G. Next, our RNA will be used to generate protein through translation. And remember that during this process, RNA nucleotides are read in groups of 3, called codons, in order to generate corresponding amino acids. Now, just very generally, we say that mutations have the effect of making this synthesized protein not turn out quite right. So I'm going to give a quick shout out to sickle cell disease, which is an example of a disease that's caused by a genetic mutation. So you may remember that there is a protein in red blood cells called hemoglobin, which we can also call HB. And hemoglobin is a protein that coordinates to iron ions in order to hold on to oxygen molecules and transport them throughout the body. Now, the mutation that causes sickle cell disease results in a mutated form of hemoglobin called HbS being formed, where the S is for the word sickle. And the difference between normal hemoglobin and HbS is that one glutamate amino acid residue is being replaced with a valine amino acid residue. And this small change results in all of these mutated HbS proteins aggregating together in a red blood cell, which makes it very difficult for that red blood cell to transport oxygen effectively. Now, just a side point, remember that red blood cells are initially generated from hematopoietic stem cells through a process called hematopoiesis. So where are mutations found, and how did they come up in the first place? Well, let's look at a couple different possible mistakes that could lead to an incorrectly produced protein. So first, we'll see what happens if a cell makes a mistake during translation, and we'll stick with our example of sickle cell disease from before. So let's say that we have this sample piece of DNA with 3 nucleotides from the gene coding for hemoglobin. This DNA is transcribed to form the complementary RNA sequence GAG. Now that GAG would normally correspond to a glutamate residue during translation, but a mistake during translation might lead to a valine residue being translated instead to produce the mutated hemoglobin associated with sickle cell disease. But notice that if a mutation happens during translation, the cell will only produce one mutated hemoglobin, or HbS, for each overall mistake. And since cells are making tons and tons of hemoglobin, just one mutated protein might not have that big of an effect on the cell. So we can say that mistakes during translation probably don't cause mutations like the one associated with sickle cell disease. So next, we'll look at mistakes during transcription. Again, we have our CTC piece of DNA, which would normally make GAG on RNA, but maybe a mistake occurs which leads to the transcription of a GUG instead, which would then code for the valine associated with mutated hemoglobin. Now, if this mistake occurred, the cell would only make a few mutated hemoglobins for each mistake, since an individual strand of messenger RNA will only be translated a couple of times before being degraded. So we can say that mistakes during transcription probably don't cause mutations like the one associated with sickle cell disease. Finally, we'll look at mistakes in the DNA strand. If our CTC in DNA is mistakenly turned into a CAC, then our corresponding RNA from transcription will be changed, and ultimately a valine will be produced instead of a glutamic acid. Now, since a cell's DNA stores all of its genetic information, that mistake would lead to all future hemoglobins produced from that gene being mutated. So overall, we can say that mutations will usually result from mistakes in a cell's DNA, and not from the RNA or the protein. So where do these types of mutations come from? Well, there are 2 ways a person can get a genetic mutation. The first is that they inherit it from their parents. Remember that DNA is passed down from parents to offspring, so if we have a mutated father here, then there's a good chance that at least one of his kids will inherit that mutated gene the same way that the child might inherit any amount of that parent's DNA. The other possibility is that the mutation will come on spontaneously, which is where a person suddenly gets a mutation in their DNA without their parents having had the same mutation. And spontaneous mutations can come from many different sources, with just a few examples being from DNA replication errors, environmental factors like certain poisons, or it's also possible that genetic mutations can come on entirely randomly. So what did we learn? Well, first we learned that mutations originate at the DNA level, and not at the RNA or protein level. But the effects of a mutation, like the example we gave with sickle cell disease, are found with problems with the proteins that are ultimately expressed by the mutated DNA. Now, like every rule, there are a couple of exceptions to this one, but we can say that the effects of a mutation are usually found at the protein level. And finally, we learned that mutations are either inherited from a parent or come on entirely spontaneously.